Let's start number 10. The Yamal anthrax outbreak. August 2016. The Yamal Peninsula, Siberia, deep inside the Russian Arctic. A heat wave has pushed temperatures to nearly 35 degrees Celsius, more than 10 degrees above the seasonal average. And the permafrost that has stayed frozen solid for decades is starting to thaw in places it never has before. On a remote reindeer pasture near the Ob River, herders begin finding animals dead in the grass with no obvious cause. Within days, more than 2,300 reindeer are dead. A 12-year-old boy named Dennis Kokorin, hospitalized with a raging fever, becomes the first confirmed human case. He dies. 90 more people are hospitalized. Investigators trace the outbreak to a single source: the exposed carcass of a reindeer that died of anthrax roughly 75 years earlier, its frozen body acting as a kind of biological time capsule the whole time. Anthrax spores, unlike most bacteria, do not simply die when their host does. Bacillus anthracis can form a dormant, armored spore capable of surviving in soil for what researchers conservatively estimate at 70 years, and some microbiologists argue considerably longer, given the right conditions of cold and low oxygen. The last confirmed anthrax outbreak on the Yamal Peninsula before 2016 was in 1941. For 75 years, nothing happened. Then a heat wave thawed ground that had been frozen since the Stalin era, and the spores that had been waiting the entire time simply picked up where they left off. Here is the detail that has never been fully resolved. Russian scientists estimate that roughly 0 reindeer carcasses from anthrax outbreaks over the past century and a half are buried in shallow informal graves across the Yamal and neighboring peninsulas, not in the deep sealed cattle burial grounds used in Western countries, but in the permafrost itself, which for most of the 20th century was assumed to be permanent. A 2011 study published in the journal Global Change Biology, led by researchers including Boris Revich of the Russian Academy of Sciences, warned specifically that continued Arctic warming could reactivate these old burial sites years before the actual outbreak happened. Nobody acted on the warning in time. Russian authorities responded by airlifting in military biological defense units and vaccinating roughly 250,000 reindeer across the region within weeks, a scale of emergency response that itself signals how seriously the outbreak was taken once officials understood what they were actually dealing with. Epidemiologists brought in to investigate noted something else troubling in the case files. Denny Kokorin's death and the 90 hospitalizations were not spread evenly across the affected zone, but clustered tightly around families who had been in direct contact with the exposed carcass or its meltwater runoff. A transmission pattern consistent with an unusually concentrated, high-dose exposure event rather than a slow, diffuse outbreak. That concentration is itself part of what makes permafrost-sourced anthrax different from the anthrax public health officials are used to managing. Modern anthrax cases, rare as they are, typically involve isolated animal contact. A single thawed carcass, releasing decades of accumulated spores into groundwater and grazing land all at once, is a different kind of event entirely, and one epidemiologist had almost no historical precedent for modeling in advance. What makes this case genuinely unresolved rather than a closed historical curiosity is the math. The Arctic is warming roughly 4 times faster than the global average, according to a 2022 study in the journal Communications, Earth and Environment. Permafrost across Siberia, Alaska, and northern Canada is thawing at rates that were not expected until the 2050s. Every thawed centimeter is a chance for something old to surface again. The 2016 outbreak was contained within weeks through mass vaccination of reindeer herds and evacuation of affected families. But researchers at the Vector Institute, Russia's State Virology and Microbiology Research Center, have publicly stated that the exact number and location of historical anthrax burial sites across the Russian Arctic is not fully mapped, meaning nobody can say with confidence how many more of these frozen graves exist, or when the next heat wave finds one. Nobody has closed the file. Before we uncover the next forgotten pathogen, make sure to subscribe. We dive deep into real mysteries, hidden history, and the science behind humanity's darkest outbreaks. If that's your kind of content, hit subscribe now. You won't want to miss what's coming next. Number 9. Smallpox in the Siberian permafrost mummies. Smallpox, caused by the variola virus, killed an estimated 300 million people in the 20th century alone before a global vaccination campaign led by the World Health Organization declared it eradicated in 1980, the only human disease ever formally eliminated from nature. Two known laboratory stockpiles remain, one in the United States, one in Russia, both kept under extraordinary biosecurity, both the subject of decades of unresolved debate about whether they should finally be destroyed. That is the official story. What complicates it is what keeps turning up in the ground. In 2004, a team of Russian scientists, led by virologist Alyssa Krantz, published findings on cadavers exhumed from a mass smallpox grave in Yakutia, the same permafrost region where mammoth carcasses regularly surface after thousands of years. The bodies dated to a 19th century smallpox epidemic, and DNA fragments of the variola virus were recovered from the frozen tissue. The fragments were not infectious, not intact enough to replicate, but their presence proved something unsettling. The genetic material of a supposedly eradicated virus can persist preserved inside permafrost far longer than public health officials had assumed when eradication was declared in 1980. The concern sharpened considerably in 2012 when a team led by Russian researchers examining a different set of remains near the Kolyma River, also victims of a 19th century epidemic, reported viral DNA fragments consistent with variola in tissue samples taken from bodies preserved in permafrost for well over a century. A 2016 assessment commissioned by the World Health Organization's Advisory Committee on variola virus research reviewed the risk and concluded that fully infectious smallpox virus surviving in permafrost mummies was, in their words, unlikely but could not be completely excluded. A carefully hedged sentence that satisfied almost nobody on either side of the debate. Virologists who study poxvirus stability point out that variola is a relatively hardy DNA virus that has been shown in laboratory conditions to remain viable in dried scab material for years under the right temperature and humidity. Nobody has ever recovered a fully infectious sample from a frozen 19th century body and can say for certain how long that viability window actually extends in the ground. So here is where the smallpox permafrost question sits today. Every burial ground from every 18th and 19th century Siberian smallpox epidemic has not been mapped, cataloged, or systematically tested. Climate change is actively eroding riverbanks and thawing ground across exactly the region where these epidemics were most severe, occasionally washing entire burial sites into rivers before anyone can study them. The virus that humanity spent 2 centuries eradicating from the living world may still be sitting dormant in the dead. Nobody knows how many graves are out there. Nobody has finished checking number 8, the resurrected 1918 influenza virus. The 1918 influenza pandemic killed an estimated 50 to 100 million people worldwide in roughly 15 months, more than died in the entirety of World War I, which had just ended when the second and most lethal wave of the pandemic hit. For most of the 20th century, the actual virus responsible was gone, a genetic ghost known only through historical description, because influenza viruses degrade quickly outside living tissue and nobody had preserved a usable sample. Then, in 1997, pathologist Jeffrey Taubenberger of the Armed Forces Institute of Pathology began recovering fragments of viral RNA from preserved lung tissue of soldiers who had died of the 1918 flu, samples that had sat in a government archive for 8 decades. In 2005, after years of painstaking genetic reconstruction, Taubenberger's team, working with researchers including Terence Tumpey at the Centers for Disease Control, successfully rebuilt the complete genome of the 1918 virus and, in a biosafety level 3 laboratory under extraordinary containment protocols, brought it back to life. The results, published in the journal Science, confirmed the virus was an H1N1 strain of avian origin that had adapted with terrifying efficiency to infect human lung tissue. Tests on mice showed the reconstructed 1918 virus was significantly more lethal than any modern flu strain used for comparison. Here is what has never been resolved. One of the most crucial pieces of tissue used in the reconstruction came from the frozen body of an Inuit woman buried in permafrost near Brevig Mission, Alaska, exhumed in 1997 by a team that included retired pathologist Johann Hulten, who had first attempted the same exhumation, unsuccessfully, back in 1951. Nearly 90% of the village's adult population had died within 5 days of the 1918 outbreak reaching it, and mass graves were dug into permafrost that has remained frozen ever since, preserving lung tissue well enough to eventually rebuild a virus that had otherwise vanished from the earth. The same permafrost condition that made the reconstruction scientifically possible is now retreating across Alaska at a rate that has alarmed climate researchers, and virologists have never conducted a comprehensive survey of how many other 1918 mass graves sit in similarly thawing ground across Alaska, Canada, and Siberia, or what condition the viral material inside them is actually in as the ground around them destabilizes. The reconstructed virus itself remains stored, alive, under some of the strictest biocontainment protocols on Earth, a decision that itself generated fierce and unresolved scientific debate after 2005, with some virologists arguing the reconstruction provided invaluable insight into what made the strain so lethal, and others, including prominent biosecurity researchers, arguing that recreating one of history's deadliest pathogens and publishing its complete genetic blueprint was an unnecessary risk that can never be fully undone. The genome sequence is now public. The file on whether that was the right call has never closed. Number 7. The Justinianic Plague's ancient strain. In the year 541, the Byzantine port city of Pelusium in Egypt became ground zero for what the contemporary historian Procopius described as a pestilence that, in his words, embraced the entire world. The Justinianic Plague, named for the Byzantine emperor Justinian I, who reportedly caught it and survived, went on to kill an estimated 25 to 50 million people across the Mediterranean and Near East over the following 2 centuries, in outbreaks that recurred roughly every generation. For 14 centuries, historians debated what the disease actually was, since Procopius's descriptions of swellings in the groin and armpit sounded like bubonic plague, but 6th century medical writing was too imprecise to confirm it. In 2013, a team led by geneticist Michael McCormick and molecular biologist David Wagner extracted ancient DNA from the teeth of 2 skeletons buried in a mass grave near the Bavarian village of Aschheim, Germany, dated to the mid-6th century through the burial's associated grave goods. The results, published in the Lancet Infectious Diseases, confirmed the presence of Yersinia pestis, the same bacterium responsible for the medieval Black Death 8 centuries later. The 1,400-year-old case was finally closed. Except the genetic sequencing opened a stranger one. The strain of Yersinia pestis, recovered from Aschheim, turned out to be genetically distinct from the strain that caused the Black Death in the 14th century, and distinct again from the strain responsible for the modern plague pandemic that began in China in the 1890s. This meant the Justinianic strain represented an entirely separate, previously unknown branch of the plague bacterium's evolutionary tree, one that appears to have gone extinct, vanishing from the historical and genetic record after roughly 2 centuries of recurring outbreaks. Nobody has been able to fully explain why. A leading theory, advanced by evolutionary microbiologists studying the Aschheim genome, is that certain genetic mutations that made the Justinianic strain so severe may also have made it evolutionarily unstable, burning through susceptible rodent and flea populations faster than it could sustain itself, effectively killing off its own transmission chain. Other researchers argue the sample size is simply too small, drawn from a handful of skeletons in one German grave, to confidently characterize an entire pandemic strain's evolutionary fate across the whole of the Mediterranean world. What nobody disputes is this: Yersinia pestis has resurfaced as an entirely new pandemic strain at least 3 separate times in recorded history, each with a genome different enough from the last that researchers initially could not confirm they were looking at the same disease. The bacterium survives today in wild rodent populations across parts of Asia, Africa, and the American Southwest, with the World Health Organization recording several thousand cases of plague worldwide every year in the modern era. A pathogen that has already reinvented itself 3 times, each time from reservoirs nobody fully mapped in advance, leaves open a question evolutionary microbiologists have never been able to answer with certainty. What stops a fourth reinvention? Number 6. The prehistoric giant viruses of the permafrost. In 2014, a team led by French virologists Jean-Michel Claverie and Chantal Abergel at Aix-Marseille University extracted a sample of permafrost from the Kolyma River lowlands in far northeastern Siberia. Dated through radiocarbon analysis of surrounding organic material to more than 30,000 years old, older than the extinction of the woolly mammoth, older than the cave paintings at Lascaux. Inside the sample, the team identified an entirely new type of virus, one large enough to be visible under a standard optical microscope, a scale almost unheard of in virology. They named it Pathovirus sibericum, and when they exposed it in laboratory conditions to a culture of amoebae, it woke up and began infecting them. 30,000 years after it was frozen, Pathovirus turned out to be harmless to humans and animals, infecting only single-celled amoebae, and the discovery was treated by much of the scientific community as a genuine curiosity rather than a threat. But Claverie and Abergel did not stop there. In 2015, the same team identified a second ancient giant virus from the same permafrost core, Mollivirus sibericum, also successfully revived, also still infectious after tens of thousands of years frozen. Then, in 2023, the team pushed the timeline further, reviving several additional strains of ancient viruses, including one dated to nearly 48,500 years old, the oldest virus ever revived by science, recovered from permafrost sampled beneath a Siberian lake. Here is the detail Claverie and Abergel have repeatedly emphasized in interviews and in their published research, and it is the part that has never been resolved. Every giant virus they have successfully revived so far happens to infect only amoebae, which the researchers themselves describe partly as a matter of what they specifically went looking for using amoeba cultures as bait, because amoeba-infecting viruses are easier and safer to isolate and study. Their stated concern, published explicitly in a 2023 paper in the journal Viruses, is that the same permafrost conditions capable of preserving an infectious 500-year-old amoeba virus are, by definition, also capable of preserving other pathogens, including ones that infect humans, and that industrial mining and oil drilling operations now actively underway across previously undisturbed deep permafrost in Siberia could expose ancient microbial material that has had no contact with any living immune system in tens of thousands of years. Nobody has systematically surveyed what else is down there. The amoeba viruses were simply the ones scientists happened to find first, using a method designed to find exactly that kind of virus. What a method designed to look for human pathogens in the same soil might turn up has never been tested. There is a second, quieter piece of this story that rarely makes it into headlines about the giant viruses. Claverie and Abergel samples were taken from a specific, relatively shallow layer of permafrost near a lake in Yakutia. The deeper layers of Siberian permafrost, some dated to over 1 million years old in places, remain almost entirely unsampled for viral content, because drilling and safely transporting deep ice cores from remote Arctic terrain is logistically difficult and extraordinarily expensive, and because until Pathovirus turned up alive in 2014, most virologists assumed nothing viable could possibly survive that long anyway. That assumption has now been overturned twice, by Mollivirus and then by the 500-year-old strain recovered in 2023, and each time the working assumption about the outer limit of viral survival in ice has simply moved further back. Claverie has said in interviews that he sees no obvious biological reason the same preservation mechanism would not extend considerably further, potentially into permafrost layers that have sat frozen since before modern humans existed as a species at all. Number 5. The ancestral strain behind the Black Death. In October 2011, a team led by geneticists Kirsten Boz and Hendrik Pinar at McMaster University, working with researchers from the University of Tübingen, published the complete genome of Yersinia pestis, recovered from the teeth of skeletons buried in East Smithfield, London, a mass burial ground used specifically for Black Death victims between 1348 and 1349, at the height of the pandemic that killed an estimated 75 to 200 million people across Europe and Asia. Somewhere between 30 and 60% of the entire population of the continent in under a decade. The genome, published in the journal Nature, confirmed definitively that Yersinia pestis caused the medieval Black Death, settling a controversy that had simmered for decades among historians who had proposed alternative culprits, including anthrax and a hemorrhagic viral disease similar to Ebola, on the grounds that the speed and pattern of the Black Death spread seemed, to some scholars, inconsistent with a flea-borne bacterial disease. The genetic evidence closed that debate. It also opened a new one, because Boz and Poinar's team found that the 14th century strain sat at what they described as a genetic node, essentially the common ancestor of every single strain of plague that exists anywhere in the world today, including the strains still circulating in wild rodent populations in Madagascar, the American Southwest, and Central Asia right now. That finding matters because of what a follow-up study did with it. In 2016, a team including Kirsten Boz, examining plague genomes recovered from later medieval burial sites, found that the Black Death's ancestral strain continued mutating and re-emerging in distinct waves across Europe for the following 4 centuries, through the Great Plague of London in 1665 and beyond, each new wave genetically related but not identical to the last, evidence that the bacterium was not simply lingering unchanged, but actively continuing to evolve inside European rodent and flea populations long after the initial catastrophic pandemic. Modern strains of Yersinia pestis, genetically speaking, are direct descendants of the exact same lineage that emptied medieval European cities. The World Health Organization currently classifies plague as a re-emerging disease, citing periodic outbreaks in Democratic Republic of Congo, Madagascar, and Peru in recent years, some involving the more dangerous pneumonic form that spreads person to person through respiratory droplets rather than flea bites. Antibiotic treatment exists and is effective when administered early. What has never been fully resolved is a question raised explicitly by Boz and Poinar's own research. A bacterium capable of persisting, mutating, and re-emerging in devastating new forms across nearly 7 centuries of recorded history offers no particular scientific reason to assume that process has stopped. Number 4. Ancient tuberculosis and the seal connection nobody expected. Tuberculosis is often described as an old world disease, assumed by generations of archaeologists to have arrived in the Americas only after European contact in 1492, carried by colonizers to populations with no prior exposure and therefore no immunity, a standard and largely uncontested historical narrative for over a century. Then, in 2014, a team led by Kirsten Boz and Johannes Krause examined skeletal remains from a pre-Columbian burial site in southern Peru, showing clear pathological evidence of tuberculosis-like bone lesions in bodies radiocarbon dated to roughly 1,000 years before Columbus ever reached the Americas. The genome the team recovered, published in the journal Nature, confirmed the bacterium was genuinely Mycobacterium tuberculosis, a member of the same bacterial complex responsible for the disease today. What nobody expected was which branch of the tuberculosis family tree it belonged to. The strain infecting pre-Columbian Peruvians was most closely related, genetically, not to any human tuberculosis strain found in Europe, Asia, or Africa, but to a strain of tuberculosis that today infects seals and sea lions along the coasts of South America and Southern Africa. The implication, laid out by Boz and Krause's team, is that tuberculosis reached the pre-Columbian Americas not overland via ancient human migration across the Bering Land Bridge, as had long been assumed for any disease present before 1492, but across the open ocean, carried by infected marine mammals that pre-Columbian coastal populations hunted, butchered, and lived alongside. The finding rewired the accepted history of one of humanity's oldest and deadliest diseases. Tuberculosis still kills more people worldwide every year than any other single infectious disease, according to World Health Organization figures. And it did so by exposing a transmission pathway nobody had previously considered central to the bacterium's global spread. Not human to human first, but animal to human, repeatedly, independently, across species and oceans, in ways the fossil and genetic record had simply never been examined closely enough to reveal. What remains genuinely unresolved is how many other independent animal-to-human tuberculosis jumps happened in prehistory that left no clear trace, and whether the modern strains driving today's global tuberculosis burden, including increasingly antibiotic-resistant strains, now tracked with growing alarm by public health agencies, share more of that ancient zoonotic ancestry than current classification systems account for. Boz and Krause's own paper is explicit that the pre-Columbian Peruvian strain itself appears to be extinct, replaced entirely by post-contact European strains. But the mechanism it revealed, ancient disease crossing from marine mammals into humans without warning, is not a closed historical event. It is a pathway biologists studying modern zoonotic spillover say could, in principle, happen again. Number 3. The Antonine Plague and the pathogen nobody has identified. In the year 165, Roman legions returning from a campaign in Mesopotamia brought home more than victory. Within months, a pestilence tore through the Roman Empire with a severity ancient physicians had never documented before. The Greek physician Galen, who personally treated patients during the outbreak, left detailed clinical descriptions: fever, diarrhea, a rash covering the entire body that turned black in places and eventually flaked off, symptoms severe enough that Galen fled Rome for his home region rather than continue treating cases, a decision historians have debated the ethics of for centuries. The Antonine Plague, named for the reigning emperor Marcus Aurelius, is estimated by historians, including demographer Walter Scheidel, to have killed somewhere between 7 and 10 million people, roughly a quarter of the population in some of the hardest-hit regions of the Roman Empire, over roughly 2 decades of recurring waves. For nearly 900 years, what actually caused it remained a matter of educated guesswork built entirely on Galen's clinical notes. Based on the described rash fever pattern and the disease's evident severity, the leading modern hypothesis, argued by historians of medicine including R.J. Littman and M.L. Littman in a widely cited 1973 analysis, is smallpox. Measles has also been proposed, on the grounds that measles and smallpox were harder to clinically distinguish in antiquity than they are today, and that measles is known to have caused catastrophic mortality in populations encountering it for the first time. A third hypothesis, favored by some paleopathologists, holds that the Antonine Plague may represent an entirely different, now extinct viral disease with no modern equivalent at all. Here is why the case has never closed. Unlike the Justinianic Plague or the Black Death, no ancient DNA confirming the Antonine Plague's cause has ever been recovered, because no mass grave definitively linked to the outbreak and containing well-preserved organic material has ever been excavated and successfully sequenced. Every proposed identification rests entirely on Galen's 900-year-old clinical description, filtered through centuries of translation and medical reinterpretation compared against modern disease symptomatology. A parallel and even older case, the Plague of Athens in 430 BC, described in comparable clinical detail by the historian Thucydides, has generated an even longer list of competing diagnoses over the centuries, including typhoid fever, typhus, smallpox, measles, and, following a controversial 2006 dental pulp study by Greek researchers examining remains from a mass grave in the Kerameikos Cemetery of Athens, typhoid fever again, or a conclusion immediately disputed by other paleomicrobiologists who argued the study's methodology could not rule out modern contamination of the ancient samples. Neither the Antonine Plague nor the Plague of Athens has ever been conclusively matched to a specific pathogen using modern genetic evidence. Two of history's most devastating documented pandemics remain, in the strictest scientific sense, unsolved. Whatever they were killed millions of people twice, in 2 different centuries, without ever telling anyone its name, and nothing currently rules out the same unidentified pathogen, or its modern descendant, still circulating somewhere in a reservoir nobody has connected to it. Number 2, the Bronze Age plague that predates fleas. In 2015, a team led by geneticist Simon Rasmussen at the Technical University of Denmark, working with Eske Willerslev's Ancient DNA Research Group, screened the teeth of 101 Bronze Age skeletons from burial sites across Europe and Asia for traces of ancient pathogens, buried among ordinary Bronze Age graves, dating back as far as 5,000 years. The team found Yersinia pestis DNA in 7 individuals, the oldest confirmed cases of plague infection in human history at the time of publication, predating the earliest previously known cases by more than 3,000 years. The genome sequencing, published in the journal Cell, revealed something that upended a basic assumption virtually every historian and epidemiologist had made about how plague spreads. Every later outbreak of Yersinia pestis, the Justinianic Plague, the Black Death, the modern pandemic, spreads primarily through fleas that infest rats and other rodents, a transmission mechanism that depends on a specific gene allowing the bacterium to survive and multiply inside a flea's gut, ultimately blocking it so the starving, infected flea bites more aggressively and transmits the bacterium more efficiently. The Bronze Age strains Rasmussen's team recovered lacked that gene entirely. This version of the plague bacterium, circulating across Bronze Age Europe and Asia for at least 2 millennia before the gene mutation that enabled flea transmission ever appeared, could not have spread the way every later plague pandemic did. It had no flea vector. It had to have been transmitted some other way, most plausibly, according to Rasmussen's team, through direct respiratory contact between infected individuals, essentially airborne, in a form of plague considerably more like modern pneumonic plague, one of the deadliest and least treatable forms of the disease, if not caught within hours. What has never been resolved is exactly how a bacterium that spread for millennia without ever needing a rodent flea vector eventually acquired one, and what pressure caused that fundamental transition, sometime between the Bronze Age strains and the earliest confirmed flea-transmissible strains identified in later Iron Age burials. A 2018 follow-up study led by Rasmussen's team identified even earlier plague genomes, pushing the earliest confirmed case back to nearly 5,700 years ago in remains from present-day Latvia, deepening the mystery of the bacterium's original transmission method without resolving it. Modern Yersinia pestis retains the capacity for both transmission routes, flea-borne bubonic plague and directly transmissible pneumonic plague, and the World Health Organization continues to record pneumonic plague cases, including a cluster in Madagascar in 2017 that infected over 2,000 people in a matter of weeks, specifically because of its capacity for direct person-to-person spread. The ancestral, flea-independent form of the disease that killed people across Bronze Age Eurasia, for 2,000 years before rodents were ever part of the equation, has never entirely gone away. It simply learned a second way to travel. And modern medicine has never fully explained what circumstances taught it the first one, or whether the underlying respiratory transmission capability the Bronze Age strains relied on ever really left the bacterium's genetic toolkit at all. Number 1. The Deep Permafrost's Unmapped Microbial Archive. In 2005, a team led by NASA astrobiologist Richard Hoover and microbiologist Elena Rogojina examined a sample of Siberian permafrost recovered from a depth of roughly 30 meters, dated to approximately 8 million years old, well beyond human history entirely, into deep evolutionary time. Inside it, they identified viable, culturable bacteria, organisms that had remained in a dormant, low metabolism state for millions of years and could still be revived and grown in a laboratory dish. The finding, presented at a NASA astrobiology conference and later expanded in peer-reviewed research, was framed at the time primarily as evidence for how life might survive on frozen worlds like Mars or Jupiter's moon Europa. It also quietly established something with far more immediate implications closer to home. Permafrost is not simply a freezer that kills everything it touches. For certain hardy microorganisms, it is closer to a waiting room. Since that discovery, the pace of confirmed ancient organism revivals has only accelerated. In 2021, a team led by Jiping Zhang at Ohio State University recovered 33 distinct groups of viruses from Tibetan Plateau glacial ice cores, dated to roughly 15,000 years old, most of them entirely unknown to modern science, belonging to no previously cataloged viral family. In 2023, French researchers Claverie and Abergel, the same team behind Pithovirus and Mollivirus, published a formal review in the journal Viruses, cataloging every giant virus successfully revived from permafrost to date, and explicitly warning that the scientific community has, in their words, no comprehensive inventory of what pathogens, bacterial, viral, or otherwise, actually exist within the vast stretches of permafrost, now beginning to thaw across an area covering roughly a quarter of the entire land surface of the northern hemisphere. Here is the part that has never been resolved and, by the nature of the problem, cannot easily be resolved through any single study. Permafrost across Siberia, Alaska, and northern Canada holds an estimated 1 in 400 to 1,600 billion metric tons of frozen organic carbon. According to a 2019 assessment published in the journal Nature Reviews Earth and Environment, a figure that gives some sense of the sheer biological scale of material locked inside ground that, in some regions, has remained continuously frozen for hundreds of thousands of years. Within that mass sits an unknown quantity of ancient microorganisms, some studied, most not, because systematically surveying frozen ground at that scale, across some of the most remote and inaccessible terrain on the planet, has never been attempted and, by most estimates from permafrost researchers, would take decades even with unlimited funding. Meanwhile, industrial activity, oil extraction, mining, and infrastructure development, driven partly by the same warming that is thawing the ground in the first place, is actively disturbing deep permafrost layers across Siberia that had never previously been exposed to open air or living organisms with modern immune systems. A 2021 modeling study published in the journal PLOS Computational Biology, led by ecologist Corey Solomon, took a different approach to the same problem. Rather than trying to physically sample every stretch of permafrost, the team simulated what happens when frozen viral communities are released into new environments after thousands of years of isolation, and found that a small but consistent percentage of ancient, previously unencountered viruses successfully infected host organisms they were exposed to in the simulation, occasionally with more severe effects than viruses the host species had prior evolutionary experience with. The study's authors were careful to note their model could not predict specific outbreaks and was not a forecast of any particular pathogen. What it did demonstrate, in the researchers' own words, is that the biological plausibility of an ancient pathogen successfully infecting a modern host after long-term isolation is not negligible, and grows with the sheer number of exposure events, which is precisely what accelerating permafrost thaw is now producing across an area covering roughly 9 million square kilometers. Nobody knows what is actually down there. Nobody has the capacity to check all of it before the thaw reaches it on its own. Every case on this list, the Yamal anthrax, the smallpox mummies, the resurrected 1918 flu, the Justinianic strain, the giant amoeba viruses, the Black Death's ancestral genome, the seal tuberculosis, the unnamed Antonine pathogen, the flea-independent Bronze Age plague, is a single confirmed data point pulled from ground that scientists happen to look at. The honest, uncomfortable conclusion researchers across every one of these fields keep arriving at independently is the same one. The confirmed cases are not the whole picture. They are simply the ones somebody happened to find. So, that was the list. The Yamal anthrax outbreak, a 75-year-old carcass that woke up the moment the ground finally warmed enough to let it. The smallpox permafrost mummies, an eradicated disease's genetic ghost still sitting in frozen 19th century graves nobody has fully mapped. The resurrected 1918 influenza virus, rebuilt from a body in Alaskan permafrost, and now stored alive under maximum containment. The Justinianic Plague's lost genetic branch, a strain of Yersinia pestis that vanished from history after 2 centuries and has never been fully explained. The giant viruses of the deep permafrost, infectious after 48,500 years, found, using a method that was never designed to look for anything that infects humans. The ancestral Black Death genome, sitting at the root of every plague strain alive today. The pre-Columbian tuberculosis strain that crossed an ocean inside a seal. The Antonine Plague and the Plague of Athens, 2 of history's deadliest pandemics that modern science has never been able to name. The Bronze Age plague that spread for 2,000 years with no flea vector at all. And the deep permafrost's unmapped microbial archive, holding an unknown quantity of ancient pathogens beneath roughly a quarter of the entire northern hemisphere's land surface, thawing faster every single year. Did we get the order right? Should the deep permafrost archive sit lower on the list, since nothing inside it has actually caused an outbreak yet? Should the Yamal anthrax case be number 1, since it is the only entry on this list that has already, in living memory, killed someone? Drop a comment right now and tell us your ranking. Tell us which one you would have moved. Tell us where in the world you are watching this from, because this community stretches across every continent, and every single one of you is the reason this channel keeps making these deep dives possible. If this list changed the way you think about what is actually sitting in the ground beneath our feet, smash that like button. It tells YouTube that real science deserves to be recommended. Subscribe and turn on notifications so you never miss the next investigation into the outbreaks, the vanished pathogens, and the unanswered questions the ancient world left frozen behind. We have more of these coming. Thank you for watching. These 10 pathogens survived ice ages, burials, and millennia of complete isolation, made it into modern laboratories with instruments their original victims could never have imagined, and in every single case, walked out the other side of the test still capable, under the right conditions, of doing exactly what they were built to do. We'll see you in the next one.